You're not allowed to share your screen.
Okay.
Working now. Thank you.
Thank you, sir.
Swati, could you please confirm you can see the slide as full screen as well as the pointer in red?
Yes, sir, it's working.
Perfect.
Okay.
Thank you very much.
Okay. Welcome back, everyone. I hope you enjoyed the break. Now we are going to proceed with our next lecture by Dr. Muhammad Asim on the topic, the drivers of cancer. We warmly welcome him again and look forward to an engaging and thought-provoking talk. Sir, over to you.
Thank you very much. Uh, okay. Do we need to wait? I'm seeing only 14 of people have joined, 15 now. So shall we wait a couple of more minutes? Or
Yes, sir, a few minutes would be nice.
this is being recorded, right?
Yes, sir.
Okay. Uh,
Meanwhile, if anyone has any question or
yes, yes,
um,
sure. Please
okay,
feel free to in the
yes, sir.
chat box. So Khadija has Khalida, I'm sorry. She has joined, and so has Miss Shiza Siddiqui, it seems. Very good afternoon, Shiza. How are you? Brilliant. Thank you. Okay. So shall we crack on?
Yes, sir.
I think there's someone saying something, or I was hearing an echo.
So is it working fine now?
Yeah.
I think it was
You.
some background noise from my end. So
Okay.
sorry.
All right. Okay. So this is day 3 of this course, and I'm conscious that some of you are, in fact, BSc students, as we saw yesterday, and we have not a big crowd today, as we saw yesterday. So we will start, and in case there will be questions, please feel free to keep posting them in the chat box. I'll try periodically to have a look while I'm teaching and try to answer them. If not, then we can take the questions afterwards. Does that make sense?
Yes, sir.
Good. Okay, thank you. So, what we are going to learn today is about some genes. So, in subsequent lectures, we will know what is the effect of DNA damage on gene functions. We saw in the last 2 lectures, one was the damage of the DNA that occurs, and yesterday we saw what are the pathways which are there to repair the defects in the DNA. But we also learned why some cells, cancer cells, become vulnerable to drugs because they cannot really repair their DNA efficiently. So today we are going to see what sort of genes are mutated by these changes, by these agents, by natural processes, by spontaneous changes happening within a cell. So what are these genes? We're going to talk about these genes called driver genes or oncogenes today. We will see what they are. And we will learn that 95%, or shall I say over 99% of the treatments of cancer we have currently is based on targeting some sort of targets. Chemotherapy aside, which targets the DNA replication, DNA integrity, most of the targeted treatment of cancer actually are targeting a driver, which is an oncogene, as we will learn in the lecture today. So, then what we are going to discuss in the next hour is these genes called tumor suppressors. I've been calling them good genes in the past 2 days, and just to make sure that not only have faculty joining this call, but also students very early in their career ladder. So please forgive me if there is something which looks like super basic biology to you. And in addition to discussing tumor suppressor genes, we are also going to discuss how some viruses can cause cancer today. So there are genes which are known to be involved in cancer. If you take DNA out, sequence it in from patients, you will find that there are mutations, which we discussed yesterday, day before yesterday, actually, what is the definition of mutations. So the genes can be broadly classified based on their role in cancer into being proto-oncogenes, which are, should I say, ticking time bombs in a cell that can cause cancer. Then we have tumor suppressor genes. These are the good genes, which I've been mentioning in the past few lectures. And a lot, but not the least, we've spoken yesterday about genome maintenance genes. We discussed these genes, which are there to repair any defects, any threats that come to the DNA. They can actually deal with that. They can allow DNA to be repaired so that the DNA damage does not really manifest itself into a full-blown mutation. We discussed that yesterday. Any question on what we covered yesterday or so far? This is important because this will set the foundation stone for what we are going to discuss today. So if you have any confusions, please feel free to ask me any questions now, or as I said, I'm more than happy to answer any questions through the chat box. Okay, let's move on. So, what happens in cancer is you see these genes, we call them proto-oncogenes and oncogenes. I'm sure some of you might know them already. Does anyone know what do they do? Why are they there? If nature knew that these genes actually can cause cancer, why would nature give us these genes? And the rationale for this is they are needed for development. They are needed for normal growth. They are needed for proliferation. They're needed for healing, especially healing of wound. Have a look at this child here, and a few decades later, becomes this. So this is none other than myself as my childhood photo. You can see I needed those genes, proto-oncogenes, in order to grow and become what I am now. So you need these genes in order for growth. So you can see this as a necessary evil. We do need these genes in order to grow, proliferate, develop, um, progress, heal. So we have to have proto-oncogenes. The key difference is that proto-oncogenes can actually be mutated by pathways which we discussed yesterday and day before yesterday. And this mutation can cause them to become oncogene. Now, there is no 1 plus 1 equals 2 formula here. Uh, there may be different kind of mutations taking place in different part of the protein, which can cause these gain of function mutation. That is a very peculiar term. Please remember that because there will be some questions. Those of you who are attending the exam and wishing to get a certificate, is that there is a gain of function. So proto-oncogenes have their own inherent function. But on top of this, they get an anomalous and abnormal function. which causes them to not be regulated. So although proto-oncogenes can be switched on and off, as you can see from this alternator button used in some batteries, some inverters, you can switch on and off the function, uh, just like in proto-oncogenes. When they are needed to take this person from here to here, some of them may get switched on, others may not be switched on. But what happens as a result of mutation, this barrier, this regulation is abolished. And when this regulation is abolished, you can actually have a system where there is no off button left. So the genes are always on, meaning they're sending signals to grow, which is a bad news. And that is why nature cannot avoid existence of proto-oncogenes in our cells to grow, proliferate, heal, but that is a necessary evil. What is not desired is to have the mutations. And as we discussed day before yesterday, uh, we have some part to play where we can suppress the rate of mutations we get in cells through lifestyle changes. But then there are sadly parts which we cannot control, and that can lead to transformation of proto-oncogenes into oncogenes, which become switched on. And as I said, and we will discuss what is happening here, what type of mutations, what type of changes are happening in the DNA of these genes, causing them to become oncogenes. So when I said to you the gain of function mutations, which give them an abnormal function, sometimes at the expense of their regular function. So sometimes they may not be able to carry out their regular function, but 90% of the time, their primary function is over amplified. So now they're able to carry out their primary function more than what was desired. On top of this, they do not respond to any signals which were meant to block their function. In other words, they become autonomous. So, what type of mutations you might find is that leads to the gain of function is one of the primary mutations is the point mutation. And we discussed this in on day one. What are the point mutations? What can they lead to in the DNA and the alternative products that they can actually make? Then what we also discussed was oh, sorry, not discuss, but we can also have chromosomal translocations where two genes on two totally unrelated chromosomes can come together as a result of the arms of different chromosomes shifting hands, and that can lead to formation of hybrid genes. And last but not the least, amplification. So sometimes, so in a diploid cell, usually you will have two copies of genes, but what can also happen is that a particular gene is amplified multiple times. In cancer, we know some of the driver oncogenes, and I'll give you an example today, where we see sometimes a copy of the gene is not as such mutated, meaning there is no single base change, for example, or two genes fusing together, but amplification, which overall would lead to change in the sequence of the genome on that particular chromosome. But because you have lots of genes for this, lots of copies of the same gene, they're going to make a lot of the same product, which is not desired. And I'll give you an example today. Uh, as I mentioned in cancer, what happens with this. So have a look here. This is one of the classic textbook example of normal proto-oncogenes converting themselves into an oncogene as a result of mutation. And you can see here what happens. This is plasma membrane, this cartoon shows, and there is a receptor which is anchored within the plasma membrane. This is a protein. I'm not going to go into the details what sort of amino acids you will find in this region or this region, but basically this part of the receptor is the receptor binding part to the growth factors. So epithelial cells, which we discussed from majority of human cancers, they need to grow. And when they need to grow, the signal comes from another cell type. Sometimes they're called growth factors. And their binding would activate the receptor. And as a result of this, the receptor dimerizes, you can see, by binding of the growth factor, and then the internal tyrosine kinase domain, in this case, becomes active, causing signal transduction pathways to become active and allowing these cells to divide, progress into cell cycle sometimes, of course. So what happens in some mutations which happen in this scenario is that the receptor may not need the growth factors at all. In other words, they become self-sufficient. So they've lost a switch on, which is switch on will be the condition when growth factors are bound. Switch off will be when the growth factors are not bound. This is regular switch on, switch off, switch on, switch off condition, right? But what happens as a result of mutation? You can see there are mutations in some of the domains as shown by this red dots. And as a result of mutations, you can see still the receptor is able to dimerize. Here it was not dimerizing without the binding of the growth factor. So the receptor needed, in order to become dimerized and activated, it needed the growth factor. And in this case here, there is no requirement for it to have growth factor just because it has some mutations. It activates those receptor dimerization. And as a result, the signal transduction pathway that allows cells to grow, divide, proliferate, they become always active. Another example would be that there are some stop codons created at the level of DNA due to the results of mutations. And what happens here, because of these, you may actually altogether lose a part, a domain of the protein. In this case, the extracellular domain of the receptor is gone. Why is it gone? Because there are changes at the level of DNA mostly, and these changes would introduce a new stop codon before the conventional stop codon, meaning the protein is going to become shortened. This protein, unfortunately, will still be active, but it will not require a growth factor to activate it. So it will always be firing and sending signals for the cell to grow, which is of course a bad news. So both scenarios, you have switch on, switch off, switch on, switch off. But in this case, you always have this situation or this situation. They're always switched on. And that is a bad news because it is going to allow these cells to have a sustained growth promoting signal, which can actually allow these cells to start to take the first step towards growing uncontrollably. They may not form any cancer at this time. Remember what we discussed yesterday? Cancer is a multi-factorial slow process, and it requires a number of subsequent mutations after the first mutations in order to cross all these barriers to become full-blown cancer, which is able to circulate in blood, sit in new organs and grow. So this is one of the first steps to go on a cancer pathway. The example will be this mutation here, the red dot I was mentioning to you. If it is a HER2 gene, and I'll come to this gene in a minute, what is known is that there is a mutation which causes valine to glutamine point mutation. And you can see this is a HER2 receptor extracellular domain, the intracellular domain, and in a normal wild type form, there is a valine here. And what happens when the signal comes, it requires the activation through the binding of the HER2 ligand. What happens sometimes is that you can have this mutation which causes a valine to become glutamine, and this point mutation is found in breast cancer. I'll come to that in a minute as well. And as a result, what happens is that this glutamine, when valine is replaced by glutamine in this part of the receptor dimerizes and sends the growth promoting signal all the time. And that is therefore an oncogenic mutation. And this is represented by valine, so the three-letter code for amino acid val, and then an arrow and glutamine, which is GLN. And that is something that doctors, pathologists would look at. There is HER2 overexpression. And on top of this, what is happening? There are mutations. So HER2 gene itself is amplified in breast cancer. And sometimes what can happen is this can unnecessarily, these small changes can unnecessarily activate, stabilize a protein. Normally, you have proteins being formed and dying. They're being formed and they are then degraded. They're not needed. They go away when they're not needed. By having some of these mutations, they can actually escape their degradation. So they don't really die anymore. They don't and get degraded, if you understand what I mean. So what happens also in this gene is, I can tell you, this is EGF receptor, epidermal growth factor. HER2, by the way, is nothing but also an epidermal growth factor receptor. And what happened here is that as a result of mutation, you can see that the extracellular domain of this EGF receptor is gone. I was showing you this similar thing on a previous slide. And as a result of this, what happens? This was the extracellular domain, which was keeping these two um, dimers to form and become active. By losing this extracellular domain, the protein dimerizes and starts relaying a growth promoting signal as a result of it. So it's a shortened copy of the receptor. Normally, what should happen to a cell? If it produces a protein which is mutated protein, cell would degrade it, right? Other times, what will happen is what you're seeing on your screen, that these proteins still are not able, they escape the degradation, they escape being killed by a cell, because cell knows that this is not the protein which I desire to have. But in this case, what happens, it is still becoming active and leading to the continuous, unstoppable growth of cancer cells. And if I show you some real life example, have a look here. This is an immunohistochemistry slide from a woman with breast cancer on the right. This is how it looks in normal uh, cases. So normal breast epithelial cells, you can see here. If you do immunohistochemistry, which you learned yesterday, what happens is there will be some expression of HER2. Yes, because we need HER2 in order for cells to grow. Epithelial cells are one of the fastest growing cells in human body. So they will need these epithelial growth factor receptor signaling, right? They need it all the time. They are some of the fastest growing cells, as I said. Our own gut, the entire gut is replaced by some estimates within 2 or 3 years. The entire gut, we have a new gut, basically. Do we see it? No, because the cells are changing, old cells dying, new cells taking their place. Dr. Aziz can tell you about this in more precise details, but actually the whole gut is replaced. So the division is really, really quick, because they're sitting at the forefront of dealing with things, external agents, UV light. Inside our gut, we eat all kind of junk foods. And as a result, the body knows that there are more chances of having a damage, and a damage that can actually lead to mutations. So what happens here is that the HER2 is expressed in those cells as well, normal breast epithelial cells. Have a look here what is happening. You have an enormous expression by these epithelial cells, the glandular epithelial cells of the breast cancer. They're expressing, you can see from the signal, the dark brown color, there is substantially higher expression of HER2 in these cases, uh, women. So their cancer is driven by HER2. And as much as over 30% of the women will have uh, their tumors being driven by HER2 kinase overexpression. So it is being overexpressed, meaning there are more uh, protein which is being formed. There is more protein copies of HER2 in these cells, and they are sending a sustained proliferative growth division signal to these cells. And that makes actually HER2 an excellent target. Why? Because this is overexpressed, and this is overexpressed on the surface. So you can actually devise an antibody, a monoclonal antibody, as a therapeutics. So drugs don't have to be, as you know, always small molecules, chemical entities. They can be biologics as well. And an excellent example, which has been very successful in treating patients who have their tumors being driven by HER2. You can see here they respond very well to this drug called trastuzumab, or Herceptin as we call it. And Herceptin is a very successful drug, but only in a subset of patients. How big is the subset? One third of the patients. So when a patient comes to the clinic, doctor will take the biopsies, and among other things, looking for other oncogenic drivers. They will also look for drivers, for example, HER2, because a lot of women express it. And if the tumor is positive for this, they see an over representation of HER2 expression in their tumors through this IHC technique, then those women may be prescribed Herceptin, and that may actually lead to treatment getting successful. If it is given without any knowledge to a woman whether their tumor is driven by HER2 or not, the treatment is not going to work, especially if there is no HER2 overexpression, uh, which is a bad news. And that's why this is what we call personalized medicine. You need to see the underlying changes in a cancer cell, what these changes are. And on top of this, can you target those changes through a drug? This drug can be an antibody, as shown on your screen. It can be a small molecule. And in both cases, what you can do is devise treatment based on the expression. This is what we call personalized medicine. The problem, unfortunately, we face is that in this case, a lot of women express HER2. This is not the case for many cancers, as I'll come to this in a minute. You would see that there are oncogenes which are expressed in 5% tumors, in 2% tumors, in 3% tumors, and therefore it's very difficult to see which one you want to target. As Dr. Aziz yesterday alluded to, it is a moving target basically. It can change, cancer can change its reliance on some drivers at some point in their life. Later on, they may actually shift, and because they're highly plastic, the tumors are show very highly plasticity. We will see it tomorrow as well, metabolic plasticity. But because of this, it is very hard to treat, unfortunately, a disease like cancer. So what is the impact of the treatment? Women have the tumor driven by HER2. You can actually treat them with drugs like this. What about other women, two-thirds of women? So as I mentioned, one-third of the women, up to one-third of them will have a HER2 overexpression. What about the rest? They have their tumor driven by something called nuclear receptors, which we will come to in a minute. And these are called nuclear hormone receptors. But I want you to have a look at this slide showing survival of women with HER2, where the HER2 is present or not. And have a look what is happening here. You see that in chart A, this is called what we call as a survival curve, or Kaplan-Meier plots. And what you can see, the this is percentage survival on your y-axis, and this is survival in months. So you can see purple, the survival is very, very poor. If you have this particular marker, only 20% patients will be surviving after 4 years, as you can see. So let's have a look what is purple. Purple is basically HER2 negative tumors. They don't have HER2. Now you would say, hang on a minute, you were saying that HER2 is a driver. They don't have HER2. So why are they dying so enormously that only 20% surviving compared to the green one, where actually more than 50% surviving when they have HER2? I'll tell you in a minute. Have a look what is happening here with HOR, which is hormone receptor. So hormone receptors are the other uh, substantial category which drive breast cancer. And which hormone receptor? Estrogen receptor is the one that drives, and also the progesterone receptor. They are the ones that drive breast cancer. So what is happening? What is the group that is doing the worst? The group of patients. The patients who succumb to their disease are the ones who do not have HER2, who do not have hormone receptor. So sadly, these are the patients where we don't know the target. Now that slide tells you how important it is to identify targets, a drug target, to save lives of patients. You can see here HER2 negative and two hormone receptors missing. We call it triple negative breast cancer. Why triple negative? Because they don't have estrogen receptor, they do not have progesterone receptor, and they do not have HER2 receptor. So this is something which we don't understand very well. They don't have any conventional targets which we can block using drugs. On the other side of the spectrum, we have the tumors in green, and they express HER2 in women, and they also express hormone receptor. We can target HER2 by Herceptin-like drugs I showed you on the previous screen, but we can also target the hormone receptors using, for example, estrogen receptor can be targeted by drugs like tamoxifen. You can aromatase inhibitors as well, and you can see here if we know what drugs to give these patients whose tumors are driven by these drivers, the outcome of the treatment is actually very well. You can see here after 4 years, almost 50% of the women surviving. Sadly, where the tumors don't express these, there is, you can see the survival is very bleak. Only 20% women are surviving. And if you see other charts, they actually tell you a pretty similar story here. This is breast cancer specific survival, and you can see this is the overall survival, and that tells you how bad the graphs see how bad is it to have a metastatic disease. You can see the group doing the worst is the one where the cancer has gone into the brain. It has metastasized and it is in the brain. And you can see the patients who have the cancer going into liver, lung, bone, they're still doing better, shall I say, compared to those where the cancer has gone into brain. And this is because largely because we can actually perform surgeries. Not only that, there are agents which you can give to strengthen bone, gain strength back, like bisphosphonates and so on, that will allow to mitigate some of the side effects of metastasis. But eventually, all of these patients sadly will succumb to their disease. Okay. So sometimes what can also happen, I'm telling you, I was telling you in the previous slide that you can lose these domains, which will allow the cells to become active. Also, you can have the mutations I was showing in red dots, but there is something else which can also happen. The cell itself might gain the ability to make their own growth factor. Previously, in the previous similar chart I showed you, the signal for the growth factor was coming from a separate cell being produced elsewhere and being used here. But now these cells have become autonomous. They do not really need growth factor from a different cell to allow them to grow. They become autonomous. So you can see here in this uh, staining, this is called immunocytochemistry, or immunofluorescence actually. You can see here that there are cells within this tumor stained with antibodies that give fluorescent colors. So for example, this is the same tumor. You can see here it is expressing epidermal growth factor receptor, something like that. Yeah. And what is also happening, this very same cells are actually, if you do the staining for epidermal growth factor, the very same cells are also expressing the epidermal growth factor. So they don't really need the signal coming from a different cell type. So you might find sometimes that, oh, this cancer patient, if we do the sequencing, it looks like the epidermal growth factor protein looks normal, the gene is normal, there is nothing wrong. But have you looked at whether these cells are starting to make their own epidermal growth factor? So there may not be mutation as such in the receptor, but this time the cell gains the ability to create, to make their own growth factor, allowing it to have a sustained proliferation, which can actually lead to cancer. Okay, so what is happening here is I was mentioning to you that Herceptin targets the HER2 receptor, and then I introduce you to nuclear hormone receptors. What are they? Basically, nuclear hormone receptors are nothing but some receptors which are not present in the plasma membrane. If you close your eyes and as a biologist and mostly think about, oh, receptors, they're sitting on the cell surface. Many students do these kind of mistakes. I know full well. Basically, these receptors are receptors in this case because they receive a signal from androgens, which are testosterone, and estrogen, which are female sex hormones. And the receptor itself is not on cell surface. Think about that, why they're not on cell surface. Primary reason is the testosterone, estrogen, they are both hydrophobic. They can simply traverse through the plasma membrane. They don't need any gates, any channels, any receptor to be present to receive their signal on a cell. They simply come inside the cytosol. That's exactly where the receptor is. So in case of androgen receptor, where my lab has been studying their role in prostate cancer, the androgen receptor can cause prostate cancer. You asked me where the prostate gland is. It is situated underneath the bladder surrounding the urethra. So some older males will have problems passing urine. They'd feel that their bladder is full. And exactly what is happening is sometimes the prostate is growing uncontrollably. Is it cancer all the time? Not really. Many times it is a benign non-cancer condition, which is age-related, but sometimes you can have a diagnosis for prostate cancer as well. So we call this androgen receptor. What are the chances if you're a female to have prostate cancer? Exactly zero, because females don't have a prostate gland, as opposed to breast cancer, where males and females both can get breast cancers. Of course, the chances of breast cancers are much higher if you're a female. So let's spend next 10 minutes on studying the role of these nuclear receptors as oncogenes and as drivers of the disease. So have a look at this slide. This is a PET-CT scan of a patient. And you can see this is primary cancer, meaning the primary cancer simply means the cancer resides in the primary organ, which is the prostate gland in this case. And you can see here that if you take out the biopsies, biopsy is done to stage the tumor to know, is it just a start of tumor, or is it something which is really at the stage 3, stage 4, meaning very advanced stage where the treatment will become difficult, right? So if you take out these prostate glands and stain the epithelial cells, of course, all the tissue will be stained, and do the staining, you might find an increased staining. As you can see here in brown color, these cells are making more androgen receptor. An androgen receptor is known to drive prostate cancer. Patients who don't have the androgens, they actually never develop prostate cancer. Have a look what happens when this disease will progress. So at the moment you are seeing in this patient, there are some vertebra in the spinal cord, vertebral column. You can see the presence of tumor as shown by this PET-CT. I'll tell you tomorrow why, how this works, PET-CT scan. Have a look in the meantime what is happening here. So if the disease progresses on, despite being on a drug, you can see here the black staining, the imaging. that the cancer actually, unfortunately, has gone into bones. If you take out the bone sample, so you would ask me who would give the bone sample to if you're alive and you want to get give your bone sample. Basically, I have a friend who is an orthopedician, and he sees a lot of prostate cancer patients. So 15 years ago, I asked him naively a question: how come you see prostate cancer patients? You're a bone doctor, you're an orthopedician. And his answer was simple: because it causes excruciating pain in the bone. So sometimes they will have to do some biopsy to reduce the disease burden. So those patients, if you do biopsies and take those tissue, you're going to find that these tissues actually, they lose the glandular architecture. We know cancer has, in cancer you lose differentiation, but have a look here. Many of these cells are actually epithelial cells now, coming from bone, and they are expressing enormous amount of the androgen receptor. Meaning, as the disease progresses, the androgen receptor level goes up in cancer cells. So it is one of the major drivers of prostate cancer. And prostate cancer is not something which is restricted to only Western countries. There are lots of cases of prostate cancer in India these days. And what is believed now is that the cases are actually being diagnosed at much earlier. Previously, it was 70s and 80s and 90s. These days, you see patients coming to the clinic and having a positive diagnosis at even 45, 50 years of age. So, this androgen receptor, which drives the disease, have a look what it does in order to drive cell growth. Basically, the androgens, which are sex hormones, the testosterone, they're located, they're circulating, they can get very easily access through the plasma membrane. As I said, they are hydrophobic in nature. The receptor, therefore, does not need to be a plasma membrane. The receptor in this case is cytosolic. What it is doing in cytosol, it is inactive. It is considered largely inactive in cytosol. But the binding of these androgens, you can see this is cholesterolic structure, these are steroid hormones. The binding of androgens can lead to protein changing in its shape. And it happens in biochemistry all the time. You have phosphorylation, it changes the protein from becoming inactive to active. In this case, this protein is becoming active as a result of binding to a steroid hormone. Once it activates, the receptor goes into the cell. It dimerizes and binds to specific parts on the genes, driving their expression. How many genes we are talking about? Thousands of genes. Their expression is regulated by androgen receptor. So it's very difficult, as Dr. Aziz was showing other day with those charts, network analysis. The genes are being expressed. What is their overall role? That is one of the challenges to establish. You have androgen receptor driving the expression of thousands of genes. What is the downstream pathways? What is the outcome in terms of cell growth? What is the outcome for cancer? What type of pathways do they regulate? And to make matter worse, what you're seeing on your screen, androgen receptor is amplified in prostate cancer, meaning there are more copies of androgen receptor. The gene is on X chromosome. So if you're a female, you will have two copies. If you're male, you have one copy. But sometimes you see even on X chromosome, there are up to 200, 300 copies, new copies of the androgen receptor created, which will eventually lead to more protein being formed, and as a result, the androgen receptor will be more active than it should be. And that's why on a IHC scan, it would look like that these cells actually have more androgen receptor compared to normal individuals. So I will stop here, and I'm happy to take questions, Dr. Aziz.
Yes, thank you so much for setting up the post-lunch session, and we look forward to some questions from the audience.
I have to say that photo of yours, Dr. Aziz, looks like it's 10 years old. You're looking like a
Yeah,
student.
I got
Oh,
inspired
no, no,
from
that's
your photograph where I need some more growth.
not. More growth factor receptors.
Yes.
Yes, please feel free. If it is allowed, they can switch on their mics and
Yeah,
ask
they'll do
questions.
that. So
Khalida,
we
she's
can type if that is. I think the microphones are on, so they can already
okay.
do that. So in the morning session, we discussed all the techniques that are needed for multi-omics.
Okay, that's
The
good.
next generation
That's good.
sequencing and mass spec and the platforms along with the data
That will
analysis.
make my job easier in the tutorial today then.
Yeah, so they were
Thank you.
now they're at least familiar with the terms next generation sequencing, RNA-seq, and
Okay,
proteomics.
that's great.
That. So,
That's perfect. Then I can certainly try and complement that with the tutorial which I prepared.
mhm.
So if there are no questions, shall we reconvene in or
Yes,
more
we
than
can.
we have 15, 14 minutes, so feel free to ask
Uh,
questions.
I think they just 14 minutes left, so we can you can restart in three. Uh, that's
Okay,
fine.
shall we stop then?
Yeah.
Okay, all right. Thank you. See
Anybody,
you,
if anybody
everyone.
wants to ask the question, they can just type in or pick up the
Yeah,
mic.
please type it. Feel free to ask as many
We are
questions
hanging
as you
around,
like.
but we are just giving you a break to
Yeah,
absorb what
I'm
all
here as well.
Dr. Hassan has just taught us. And I've already shared the YouTube links plus the folder link. So yeah, I've got all the material there. But I can tell you that there's no replacement for a one-to-one interaction.
Ah,
So,
I agree.
so don't think that everything is available online. So I'll read it later on. It'll never happen.
You,
Ask
you
your
don't
things.
get the same. That's right. Yeah, you don't get the same understanding of a subject.
All right, Dr. Hassan. So we will be hanging around, but I'm muting now. So we will reconvene at
Sure, sure,
3 p.m.
sure. So Madiha is asking, can you please explain a little about the biochemistry behind the constitutively active receptor dimerization when valine changes to glutamine, as valine is hydrophobic amino acid and glutamine is hydrophilic. So I think I'm not a biochemist. I'm a cancer biologist, but I think what you're asking, your question has itself the answer. The changes in the so this valine is hydrophobic, and where would you find them? Normally inside the course of proteins, while the glutamine is hydrophilic. And because of this, you might actually lose you can counter the extracellular domains, keeping it apart, perhaps. So they become really, really close in the absence of, if you lose those, first of all, the extracellular domains, or if you have these mutations, they might bring the monomers into close proximity of each other. And as a result, perhaps this can lead to initiation of the signal transduction cascade. I'm sure this must have been worked out, just that I'm not completely confident how does it do so. Any other questions? Please feel free to type questions. I'll join you in 10 minutes again. I'm leaving my screen live. Shall we start, everyone? Dr. Aziz?
Yes, sir.
Yes.
Great. Okay. Thank you very much. All right. So that's where we've stopped. And there were some questions asked, some very interesting ones. Does androgen receptor always act as a oncogene or tumor suppressor as well on similar lines? So as you can see here, that androgen receptor is a driver of prostate cancer. Several years ago, and current research in my lab is trying to understand whether there are any roles of androgen receptor as being a tumor suppressor, but it largely looks like it acts as a tumor promoter by driving a gene expression programs. Which genes are being expressed by androgen receptor? You can study RNA sequencing to establish those gene sets which are driven by androgen receptor. Uh, no audio, Yusra said. Are you able to not hear? Can you hear me?
So for me, it's working fine. Can anyone
Okay.
else confirm it?
Okay. Thank you, Padmavati. Great. So we are trying to understand how these pathways are regulated. And in fact, the function of androgen receptor to serve as an oncogene versus a tumor suppressor could also be driven by whether we're talking about cancer or non-normal cells, but also the positioning of the androgen receptor. Um, this is highly confidential part of a patent, so I'm not going to talk about it, but I can offline touch base with you if you will be interested. So, because this is androgen receptor is driving this pathway, and in breast epithelial cells, what you also see is that the similar androgen receptor can drive specific forms of breast cancer, such as molecular apocrine type of breast cancer, but also estrogen receptor. So estrogen receptor will actually drive a similar signal transduction cascade in breast cancer epithelial cells to drive their growth and proliferation uncontrollably. And therefore, both androgen receptor and estrogen receptor are drug targets. They are the therapeutic targets in the clinic by drugs like enzalutamide, which can block the androgen receptor activation, and drugs like tamoxifen, which can block the estrogen receptor activation. And most of the time, these drugs are actually quite effective at curbing the tumor growth. As you can see from this PET-CT scan, there is an individual with prostate cancer, and you can see the cancer growing in, it is invading locally, as well as you can see some cancer in the bone. But you can see 2 weeks after the therapy, the cancer is looking like there is hardly any response. But have a look what happens when this person is on androgen receptor targeting therapy. The cancer, these shadows of cancer, they're largely gone. So these drugs are effective. The problem is they're effective on a time scale of uh, good few months to perhaps at best a year. Eventually they fail. So the challenge is for the next generation, for you to understand why this happens, and develop better drugs that can actually sustain in a sustained fashion block the androgen receptor activity. So this is one of the things I wanted to talk to you about. The other would be the fusion of chromosomal arms in order to give rise to hybrid genes. For example, this is what happens in a CML, which is a form of blood cancer. What you see if you take slides from these patients, immunocytochemistry slides in pathology labs, what is prominent is this presence of a new chromosome, which is otherwise not found in a regular people. This is called Philadelphia chromosome. And as a result of formation of this hybrid chromosomes, two genes come together, BCR and ABL, and that actually can drive the abnormal growth of lymphoid cells in a CML disease. BCR-ABL is going to be constitutively active, meaning it has lots of switch on and off alternator button. The BCR-ABL kinase is always active, sending the signal transduction to become always active. The cells are actively proliferating into this cell cycle, and as a result of this, manifest themselves into this form of cancer. These sort of diseases, I think, are probably much better. We are much better to treat them. Some of them are, in fact, treated, not like prostate cancer, where you have 5 months, 6 months, perhaps a year on a drug, and then the cancer comes back. In this case, sometimes the cure is really good. So the person dies of a different disease than the cancer itself. And because these genes are not present in normal cells, for example, these fusion genes, sometimes you don't really see a lot of side effects of treatments like this. Uh, so Padmavati is asking questions at 2 weeks of the therapy, the disease looks to be aggravated. You have a point certainly, what could be the reason? The reason could be that there may be a feedback switch on, or this is just an individual specific, that it takes time for cancer to start showing effect on so the molecular changes may still be happening at 2 weeks, but they are not really translated into the growth of tumors. And by the time we go into the therapy, there was no scan for 4 weeks. So perhaps you can see that if this was there, that after 4 weeks, the tumor has started to disappear. And that's missing basically from this. But have a look at this paper. The reference is provided. But very good point, Padmavati. So